Hi kiddos, sorry I'm not there today. A few things. First of all, you should have already gone over your tests, at least the multiple choice portion of it. I will be back on Monday, so I'll see you then. We need to go over 3.1, so you'll notice that there is a new folder on Schoology, and you'll want to download this PowerPoint and kind of walk through it with me. I will make sure to cover any questions, comments, concerns that you guys have on Monday when I'm back. But I just want to get you through the basics today, just so that I don't have to go over just generic vocab. Okay, here we go. Ready? Whee! Okay, so 3.1 is all about genes. Now, ladies and gentlemen, we've talked quite a bit about genes. We know that DNA has A's, T's, C's, and G's. We know that packets of that are called genes that code for a protein. So that's what you know about genes thus far. Genes are little bits of DNA that code for a protein. Okay, so you'll notice that there's 7 understandings, 2 applications, 1 skill. And that skill is using a database. And so we're going to do that next week, Tuesday. Use that database. So here is an image of a gene. I'm so sorry. That's an image of a chromosome. The whole thing is a chromosome. Each band is a gene. So you know that chromosomes are made up of DNA and proteins. The proteins are called histones. Remember, the histone is right here and the DNA wraps around the protein. And then that causes it to supercoil. Do you remember that? I hope so. So gene is something that you can inherit, so we call it an inheritable factor that controls a characteristic and has a specific location on a chromosome. Genes just aren't anywhere. They are on one specific location, and that specific location we call a locus, or you can pluralize it and call it a gene loci. So, anyway, you're going to have a chromosome. It's going to have sections of it called genes, and those genes are going to have specific locations called loci. Now, although every human has genes in the exact same location of the exact same chromosome, they might have different variations of that gene. So, if you have brown eyes, or if you have blue eyes, you both have genes that control eye color. You know that's true. But you're going to have different variations of that gene. So some of you might have a big B, if you remember those genetics from 7th grade. Or some of you might have the little b for eyes. And those different variations are called alleles. So alleles are a specific form of a gene. So, for example, eye color, like I said. Or you could talk about plant flower color, or pea pod color, or height, or weight, or anything like that. All of those would be genes that have specific alleles. So, if you look at the number of genes of a human with any other species, and this is where it gets kind of cool. So you guys know that humans have quite a few genes. We're pretty complex. On average, IB Biology likes to state that we have about 22,000 genes. So you'll notice right here on this diagram, mammals, humans, about 22,000 genes. You'll notice certain things make sense that have less genes, like birds, or specifically chickens, or insects, or bacteria and viruses, because those in our brains are less complex, therefore they should have less genes. So that kind of makes sense. However, there are times that animals or organisms that seem to us as maybe less complex actually have more genes. So let me give you an example of a grape. We have, again, 22,000 genes. So little packets of DNA that code for proteins, right? Or some sort of factor or characteristic. Grapes have 30,000 genes. So they actually have more genes, even though they seem kind of not as complex. So It's not just plants that have a large number of genes. Super simple animals like a water flea, if you want to Google that, a water flea actually has more genes than a human does as well. So there are organisms, even if they seem less complex, that have more genes. You do not need to know the specific numbers for bacteria or viruses or plants. You're just going to know in general that animals will have different numbers of genes when compared to other species. Okay? All right, so I want to watch this little supercoiling video. We've talked about supercoiling before, but I want to actually show you how this physically happens because I think it's a super neat process. So again, please remember for supercoiling, you're going to have two things. You're going to have DNA and you're going to have proteins. And those proteins are specifically called histones, okay? So here we go. Maybe. So here's our DNA. You can see the double helix, right? And then all of a sudden you're going to see these proteins swoop in. And those proteins swooping in are going to cause the DNA, there's the protein, to wrap around it. And you'll notice that a whole bunch of proteins actually join in one area to keep wrapping and wrapping and wrapping the DNA. And there'll actually be 8 histones within one little area kind of condensing. And you'll notice that they just keep condensing and condensing and condensing. And so all of a sudden we go from chromatin, if you remember, that's the spaghetti, the all uncoiled DNA, to a chromosome, which is supercoiled. So you'll notice in this animation we went from one coiling to many coiling to then many coiling on top of many coiling, and now again we're on many coiling. So it just takes that really, really long piece of DNA and scrunches it down into these chromosomal X's, right? And that's what we can actually visually see. Like when we did the cell division unit, we could visually see the chromosomes dividing between cells, right? Okay, so hopefully that helped. So, something that's really cool and something that you may not know about is this Human Genome Project. It took us about 13 years to map in a complete set of human genes from start to finish. And ladies and gentlemen, that means all of the A's, T's, C's, and G's for the entire human genome all in one giant sequence. So that's kind of cool. We'll talk a little bit more about this Human Genome Project when I get back on Monday. But anyway, as you're reading about it, you'll kind of know what it is. So you know, we started this process kind of in the mid-1970s. And at that time, scientists could really only sequence a few base pairs per year. To give you a little idea, humans have 3.2 billion base pairs. So they have 3.2 billion, with a B, A's, T's, C's, and G's that scrunch together in little packets called genes, which are coded then for proteins that are in our body, right? So we have this really big giant sequence, and little chunks of that sequence are red. Those are genes. So, by the time the Human Genome Project got into the 1990s, a few laboratories in the area had managed to start sequencing 100,000 bases or so. And that cost then was really, really, really high. So, once the big computer boom hit, and we got a whole bunch of biotechnologies like PCR, which we've talked about, it's called polymerase chain reaction, remember, makes a whole bunch of copies of DNA. And we figured out how to tag with antifluorescent dye the A's, T's, C's, and G's. And we got really cool things like digital cameras and computers that could assemble all the sequences and weren't super, super pokey. The Human Genome Project took off. And so this thing that was slowly trudging along because of technology skyrocketed and got done by 2003. So, there's a few things that I want to talk about as far as sequences and genes and amino acids that come from it. Now, ladies and gentlemen, we have done this. So we have done from DNA to RNA to amino acids. We've done that process. So, just a reminder, it's called transcription and translation. So if we look specifically on chromosome number 11, if you've ever heard of sickle cell disease, what happens is there's one change in the sequence. So CCT-GAG-GAG, there's one change in that sequence. And that actually leads to a disease on this chromosome. So if we walk through, if it were normal, please remember we use the antisense strand. We go through transcription and we make A's pair with U's now instead of T's, right? So we make that base pair. And then we would read the codon chart in the set of 3's, and we would read the amino acids, right? So if we were looking at just these 3 sequences right here, these antisense sequences, we would go to transcription, and then we'd go to the codon chart, and we'd have proline, glutamic acid, glutamic acid. And if that was the case, your red blood cells would be really nice and plump and big, and that would be ideal. They carry oxygen really well. Everything is super awesome and wonderful. Well, here's what happens. There's a mutation. So you'll notice that the A mutated and turned to a T. That causes the antisense strand to have a different letter, which causes the transcription, or the mRNA strand, to have a whole different set of codons, which then causes it to have a different amino acid. And that key piece, you'll notice that that glutamic acid that it was is now valine. And that valine has very different properties than the glutamic acid. And so what that makes it do is, instead of having this really nice plump hemoglobin, it sickles. And so you can physically see that it's kind of curved shape, like a sickle that you would use for cutting down wheat. And so it doesn't carry oxygen as effectively. So here, just looking at that glutamic acid versus valine, you can see that this has a negative. You can see that this doesn't have a negative. And so this happens to be polar, which means that it likes water, right? And it doesn't want to hide. It wants to stay out. It stays really nice and plump. This is nonpolar, which means it doesn't like water, remember. And so it wants to like curl in and hide away from the water, which isn't a good thing because then that causes it to sickle or curve. So I hope that makes sense. If it's big and nice and healthy, it carries blood, it doesn't get stuck, carries oxygen in the blood, and it doesn't get stuck anywhere. If it's sickled, it can go ahead and get stuck, causing blocking or blood flow issues. That tends to be a huge issue. So we are going to talk about the rest of this on Monday, but I wanted to make sure to just get some of the basics down. You are going to spend whatever time you have left today doing your reading notes for 3.1 and 3.2. Ladies and gentlemen, please do use your time wisely. You won't get any more work time, you know that, for your reading notes in class. So if you have any time left of the hour, please pull up the notes, get to work. Thanks so much. I'll happen to see you guys on Monday. Bye.